Are we ready?
Yes. No one here was Francesca.
So are we ready for the next speaker? Okay, so Victor Martinez from the west coast of Puerto Rico.
Hello, my name is Victor Martinez, and I'm from University of Puerto Rico, Maya West Campus, and I'm part of the Hunter College Summer Program. And the title of my project is Examining the Subcellular Localization of the Ajuba protein in Human Cells. Now, my work revolves around a cellular process known as DNA damage response. Now, there are many pathways for DNA damage response, and I will be focusing on one called the ATR pathway, which is activated by single-stranded DNA. Now, the cell elicits DNA damage response in order to prevent damage caused by these two examples, like here, UV radiation and replication stress. Now, DNA damage response is important to prevent early events of cancer. Like in the picture you can see here, you're going to have that a fully functional DNA damage response is going to prevent or inhibit cancer. But if something's wrong with the DNA damage response, it could easily lead to the development of cancerous cells. Now, what is responsible for sensing the damage? So this is where the key sensor is introduced, and it is called replication protein A. Now, RPA has the capacity to bind to single-stranded DNA in order to activate the ATR pathway. So what exactly is RPA? Now, RPA is a complex of free subunits, which has the capacity to not only bind to DNA, and it could also bind to proteins. So overall, the general function of RPA is to maintain genome stability, which is essential for the cells to survive. So here we have a summary of the DNA damage response ATR pathway. You're going to have your single-stranded DNA, and the sensor RPA is going to bind to the single-stranded DNA. So what she's going to do is she's going to recruit proteins that are vital for activation of the ATR pathway. Once the DNA damage response is activated, it's going to have some important effects. These effects could be cell cycle delay, it could be DNA repair, or programmed cell death. Now, these effects are vital for preventing, development of cancerous cells, because in cell cycle delay, it's going to prevent the division of the cells, and in programmed cell death, it's going to kill the cell, and it's going to prevent the development of a tumor or cancer cell, or more cancer cells. So, this DNA damage response is always needed. So, unwanted DNA damage response could lead to cell death. So, our previous work in our lab has shown that a protein called Ajuba interacts with RPA. Now, this interaction was proved by the technique co-immunoprecipitation. And they saw what in the previous work, they saw when Ajuba was depleted, from RPA, it leads to activation of DDR and cell death. So our model is that Ajuba interacts with RPA to prevent unwanted DNA damage response. So here is the model of the previous work. You can see here that on the left, you're going to see that DDR, ATR is inactivated. This occurs on the cell, more specifically in the nucleus during DNA replication. You're going to have that Ajuba and RPA are interacting. And why during DNA replication? Well, there's going to be a lot more accumulation of single-stranded DNA. And the single-stranded DNA, the cell can detect it has damage. So Ajuba interacts with RPA to prevent the cell from detecting single-stranded DNA has a damaged site. So if DDR and ATR is activated, you see that RPA and Ajuba are not interacting. Ajuba is totally depleted from RPA. So RPA is going to recruit all the important proteins, and you're going to have your effects from DNA damage response. So we already know that Ajuba and RPA interacts from our previous work, but we don't know is co-localization. So co-localization means is that we're going to try to find if these two biomolecules are going to overlap in some location in the cell, more specifically in the nucleus. So my hypothesis is going to be that Ajuba co-localizes with RPA in the nucleus during DNA replication. And to test this hypothesis, we're going to do co-localization between RPA and Ajuba by using the technique immunofluorescence. So with our technique immunofluorescence, we have two cell lines, or we use two cell lines. The first one is HTC75, which is fibrosarcoma tumor cells, our cancerous cells, and the other one is armor 90, which is normal diploid fibroblasts, which are normal cells. They are cells from our skin, actually. So the fluorophore used was TRIC-C for RPA, which is red, FIT-C for Ajuba green, and DAPI to stain DNA blue. On the picture you see on the right, we can see the technique of immunofluorescence we use, which is indirect. We use two primary antibodies. We use two antibodies. The primary, which is going to attach to the antigen, and the secondary is going to attach to the primary, but the secondary contains the fluorophore, which is going to be the colors you're going to see on the data. So here is the data. We're going to have our cells, which this is HTC75. You can see that the blue is going to be DAPI, which is going to stain for the DNA. The green, which is Ajuba. The red is for RPA, and the merge is the combination of the last three pictures. Now, to show that RPA and Ajuba co-localize, we inserted some little white arrows where you can see that the white arrows are pointing to dots where Ajuba and RPA are co-localizing. So in the merge, when you're going to see if they co-localize or not, you can see that it turns like a colored yellow. So the merge photo is to prove that RPA and Ajuba co-localized. The same we did with armor 90s. You can see the arrows are pointing to dots which are locations where RPA and Ajuba are co-localizing. So we found out that Ajuba partially co-localizes with RPA. We already know that they interact and they form a complex, but they partially co-localize, but only in a subset of cells. So further analysis has to be done to show co-localization between Ajuba and RPA. Now, on the graph you see below, there is a small quantification about the co-localization between Ajuba and RPA. We counted 100 nuclei for HTC75 and saw that 18% of the cells show co-localization. And we did the same for armor 90s, but we counted 50 nuclei for armor 90 and saw that 20% show co-localization. So for our future experiments, we're going to do co-immunoprecipitation, and ASCOF is Ajuba and RPA dissociate upon DNA damage, and do they co-localize in S phase. Now the picture you see below is a picture of the cell cycle, and the red arrow is pointing at the S phase, which is DNA replication phase. So we are going to treat our cells with double time eating block to arrest cells on this phase, on S phase, and we will expect an increase of co-localization between Ajuba and RPA. So here's my acknowledgements. I would like to thank my mentor, Diego Loaiza, my grad student, Sandy Fowler, and obviously Hunter College and the funding. Thank you for your attention, and if you have any questions.